My name's Sandra and I work for Blitz too, and we're here in Cape Range National Park, all the way over in Western Australia. And today with me I have Dr. Nick Titanic, who is an entomologist. Glenn, who you saw in the video, he couldn't be here this morning because he's out in the field collecting fish and doing all the exciting things. So he's sent along Dr. Mark Allen, who's also a marine scientist. And we're also really lucky enough to snag Lynette Umbrello, who for her research, she uses DNA to study populations of small mammals and how they change over time. So that's really exciting. For the last week here at Cape Range National Park, these scientists have been collecting as many species as possible to document the amazing biodiversity of this area. And at the end of each day in the field, they come back here. And this is our base camp where we stay every night. And then they look more closely at what they've been collecting. So we've got things like these big microscopes where they're looking down when they can see the tiny little specimens that they've collected. And I'll give you a quick walk around the lab. So there's lots of other scientists working here. Let's just give you a quick tour. We have Mark who's looking at spiders. And what have we got over here? Did you want to show?
Hello. Here I've got some of the crabs we caught last night when we went for a reef walk.
So there's some crabs, everyone. Cool. Thank you. All right. Thanks for sending them through. You guys had some really awesome questions. Okay, let's put you back up here so we can see everyone. So, the first question is from Joanne in 7C. Did you want to ask your question?
Joanne, I would say follow your passion in life. If you're passionate about something, chase after it, because you have to have a passion, especially as a scientist. It's a pretty hard field to get into and to get employment in, I'll be honest with you. But we're all really passionate about our jobs as scientists, and you need to have that passion. But I'll give you some practical advice as well. You might think science is all science looking down the microscope and biology or whatnot, but English and especially grammar and mathematics are super important in the science field. So if you are inclined to go down the science pathway, work really hard on your maths and your English, because it will come in very handy when you actually do go to university and if you become a scientist later on, it will help you out tremendously. So that's what I would say.
Yeah, I can add to that a little bit as well. I would say that even at your age, it's never too early to start volunteering. So if you have the opportunity to get involved with any citizen science programs or things like that, it's a really good way for you to experience what working like a scientist is like. And that will also really help later on if you go to university and then you want to get a job in a science field. You will have some connections built up already from your volunteer work. So that's something to look out for even when you're in high school, to think about if you can do some science-based volunteer work. So that would be my advice.
Great advice, guys. Thank you. So the next question, I think, is from Alyssa in 7A. Did you want to ask your question?
Okay. I'd say for me, the highlight of my career was working in the Amazon rainforest and getting to climb Amazon rainforest trees. So climbing 50 meters up off the ground and working on a documentary with David Suzuki. So when you do science, you often get to work with people who do cool TV shows and stuff. So that was probably my highlight. You guys?
So I've been really lucky. When I first started out doing my PhD research, I was doing a marine project up in a place in Indonesia called the Raja Ampat Archipelago, and it's regarded as the global centre of biodiversity for marine fauna. And it's just the most amazing coral reef environment that you could ever imagine. And I was lucky enough to go there 6 times over the course of 4 years and do many, many dives in some of the best waters in the world. And that was definitely the highlight of my career.
Great. Those are amazing experiences. Okay, so the next question from Peter in 7C.
guess we can all do it.
Yeah. So I'm a mammologist, which means I work on mammals, and the kind of taxonomy that I do involves a lot of work on DNA. So I sequence the DNA of lots of different individuals and look at how they are different all across the landscape. And I can show you guys a picture. I'll come up here. These are the little critters that I work on. So they're really teeny tiny, small carnivorous marsupials.
I'm sort of a newbie when it comes to taxonomy. My background is in ecology, where you study the interactions of animals in their environment. But my specialty is freshwater fishes and also marine fishes. So I hope to get more into taxonomy as the years go by, but I'm only just starting out, and I've only described one species so far, a little damselfish from the northwest coast of WA.
And I'm an entomologist, so I work on insects, and as you might guess, there's millions and millions of insects out there. So when you work on insects, you have to specialize on just a small group of them. So I work on true bugs, which are cute little bugs that have a straw for a mouth, and yeah, there's thousands of species out there to be discovered. So pretty cool.
Okay, and the next question is really a very, very smart one. So good one, Will. Do you want to stand up and ask your question?
With a lot of animals, depending on the group that you're working on, they're either really well known or really poorly known. So for some animals, we can actually look at published books and papers that give you keys on how you identify them. And for other groups of animals, we often don't know what they are, and we have to send them off to specialists, and they'll do some research and they'll use their experience with those animals to tell us what they might be. And the process can take anywhere from, you could pick something up and say, oh, I know what that is, or you could pick something up and 10 years later you still don't know what it is. So it really varies depending on what kind of animals you're working on.
I'll add to that. So in the museum where we work in Western Australia, in Perth, there's a really vast collection of animals there, and it's amazing. There's a lot of unidentified species in there that, like Nick was saying, sometimes there's just not enough knowledge of the group to be able to put a name on it, to classify it. So, for example, we get visitors come through our museum sometimes, specialists in certain groups that will just come and look through our collection and pull out jars off the shelf with fish in them or whatever, insects, and they'll go, oh, this is a new species, and then they'll recognise immediately that it's different, and it might have been sitting on our shelves for 30, 40 years. So it's incredible how many treasures we have at the WA Museum that are just sitting there waiting to be discovered, that have already been collected. It's kind of pretty interesting that way.
All right, thanks for that. What the next question from Sierra in 7A.
Mark kind of already answered that question a little bit just before. So after we've captured them, we might decide that we want to put them into the museum collection. And with the insects, we usually have to pin them out. So you might have seen a little bit of that when you did the tour of the lab just before. And then what we do is all of our specimens get a unique identifier number, and then we collect information about where they were captured, so the location and the date and who captured it and what the habitat was like. And that goes onto a big electronic database that keeps a track of where all our specimens go. And then we put them into the museum collection so that they can be used in the future by anyone for hopefully hundreds of years.
Awesome.
Yep.
So the next question is from Tamer in 7A.
I guess I can add to that. So Lynette already mentioned a bit about how we database all our material, and this information can become quite complex, and we end up having a network of data. We have specimens that are sampled for DNA, so they have to be linked to the specimens that are on display or that are in our collections that are dry, and then all this information also gets fed to national databases like Atlas of Living Australia is one. Yeah, and yeah, there's a few of them. So eventually I think the goal is for all museums to try and have all of our data visible to the public and visible to other researchers, so people can look at it and know where different species are distributed, and also know where potentially undescribed material might be from.
Mia from 7E, did you want to ask your question?
I'll answer that because I'm the marine person here. Not on this trip. Personally speaking, I study fishes, and there aren't a lot of recent extinctions in fish that are documented. In fact, I can't recall any, but there are a number of endangered species, mostly the shark species and some of the other relatives of sharks, rays, for example. Of course, we're in Ningaloo, or Cape Range, which is home of the one of the most famous whale shark populations, and it's an extremely busy time of year. This is the peak whale shark season here, and we haven't actually seen any, unfortunately. Most of the diving we've been doing is not in the right habitat to see whale sharks, but fingers crossed before the end of the trip, maybe we will see one. You never know. So in answer to your question, I haven't seen any, but I spoke to some of my colleagues that study at some of the other marine groups, such as the mollusks, the shells, and the sea slugs and those sorts of animals, and also the marine invertebrates. And Lisa, who's the mollusk curator here, told me that to tell you guys that she had seen a species of giant clam, which we saw on a reef walk last night at low tide. That is, it's on the endangered list, so that was an exciting find to make. So yeah, there you go. We're finding something.
Great, thanks, Matt. So Marcus from 7E, you've got the next question. Great, thanks.
So let's see. Whenever we do a bush blitz, we always find new species, and this is something that really surprises people. They think that we've described it all, that all the animals are known, but the reality is that a lot of Australia's animals and plants aren't known to science yet. So when we've gone collecting, we've only been here for about a week, and we've already got new insect species, we have new spider species, we have new nudibranchs, we have any new moths yet? We have some new moths. So yes, we've definitely got some new species here. It'll take a while for us to know what they are and to give them names, but pretty much everyone will walk home from this trip with new species, which is pretty exciting.
Okay. Great, thanks for that, Nick. The next question's an exciting one. Zahara, 7A, did you want to ask yours?
Okay. So I work on mammals, but I also work on other terrestrial vertebrates, which includes snakes and reptiles and lizards and those kinds of things. So quite often on our field trips, we will trap venomous snakes of lots of different species. So that would probably be my most dangerous is the venomous snakes.
Yep, and on this trip I haven't seen one, but a previous trip that I was partaking in 2017, we collected a reef stonefish, and they're one of the most venomous creatures in the ocean. We found it in really shallow water, so you guys are probably aware whenever you're walking along reefs or in intertidal zones along the shore in the marine environment, that there's always a possibility that you might encounter a stonefish in tropical waters. Yeah, they're seriously camouflaged, so often you might not even know they're there until you've actually stepped on one and then you're off to hospital. Luckily that didn't happen to me or any of my colleagues, because that would be seriously bad news. But yeah, they're an incredible creature, and it's probably the most dangerous thing I've ever encountered during my time in the marine environment.
Yeah, it's not dangerous.
I know.
We have one of the least dangerous animals here.
Lynette brought on something to show you guys too. So,
can you see it? Yeah. So this is just a little gecko that's been hanging around our camp. So I thought I would put him in a little terrarium so you guys could have a look. But yeah, he's pretty cute and definitely not dangerous.
okay. So in this bucket here, Mark's going to explain can you see what is it?
So the long black curled up thing around the edge of the bucket is a sea cucumber. You probably know it's a type of holothurian. It's a holothurian, which is a type of echinoderm. So it's in the same kind of group as starfishes and basket stars. I don't know too much about them, as I told you before. I'm a fish person, but and the other thing that was in there, probably be able to hold it up for you. So this is a clam, a giant clam. Doesn't look too giant, but it's a small one. And this is one of those ones that I was saying that Lisa, the mollusk curator, was really interested in because it's a species that's on the endangered list. And yeah, it was a really interesting find to get that off the reef last night. And over here, something else we found on the reef. Walking around, can you see that? It's a little baby stingray. Well, it's not a baby actually. This is probably a mature adult. This species is called Neotrigon ningaluensis, and it's the Ningaloo mask ray. So it's a species that was first discovered in this very area that we're working and described back in 2010 by scientists from the CSIRO in Tasmania. And really cute little creature, that one. It's going to go into our collection at the museum.
Perfect, thank you. Great. All right, thanks guys so much. You were awesome. And thanks so much for your exciting questions. We loved answering those for you. So yeah, keep up all the good work, and maybe one day you'll be a biologist here at Bush Blitz.
Bye-bye.